Hi, I'm Janine Cody, Director of the Chromosome 18 Clinical Research Center at UT Health San Antonio. And I'm often asked about interpreting microarray reports. So I thought I would create a little video to help people understand the report with the goal of helping you prepare for your meeting with a genetics professional who will really interpret that report. So my goal here is to help you learn some of the language in the reports, the types of information in the report, and the limits of that information. And this way, when you meet with a genetics professional, you'll really be much more prepared to understand what the implications are in the report for your family. So a little background. Here's a picture of chromosomes taken under a microscope. These are from just a blood cell. This is the old-fashioned way of doing a chromosome analysis. And the way that analysis was done was the picture was cut up and reorganized so the chromosomes are shown in pairs so you can compare each one. And they're in pairs because one comes from mom, one comes from dad. And you could easily see if you had an extra piece of chromosome or if chromosomes were rearranged some way where a chunk of one arm was swapped with a chunk of another arm of a chromosome. You could see that here. And of course, anything you could do to this picture with scissors, glue, and a copy machine can really happen, and it does really happen. And so this kind of report is still important for looking at those conformational changes where chromosome pieces swap big pieces swap places, or extra copies, and you can see where the extra pieces are, or where duplications might be located. A microarray report, on the other hand, is a molecular analysis. So the report you're going to see may have a picture like this, and you're going to know this is a molecular analysis report because these are drawings of the chromosomes. Yes, the banding pattern is there, but these drawings serve as sort of a map of the chromosome. And you know that it's for a microarray report because these chromosomes are not shown in pairs. It's just a single copy of each chromosome considered to be the map of the chromosome. And so a molecular analysis is really, really good at telling you the number of copies of various positions along the chromosome. And chromosome microarray analysis today has hundreds of thousands of points along the chromosomes that it's determining the number of copies of each of those points. So it's very high resolution. But it only tells you the number of copies at each of those locations. And that's really important because if you look at these old karyotypes, here's two different people with deletions of the end of chromosome 18, the long arm of chromosome 18. Here they are zoomed in, and you can see they kind of look about the same. But what we know from molecular analysis is they're not the same. One of these individuals has a deletion, a missing piece from the end of the chromosome. The other one has a missing piece from near the end of the chromosome, not the end of the chromosome. So in fact, they are quite different, even though they look about the same here. So this is where molecular analysis is so important to really distinguish exact locations of deletions in this case, or duplications, and aligning that with the actual genes. Here, there's no way to know which genes are involved. So it's a really, really important technique to use. And again, molecular analysis or microarray analysis just determines the number of copies at each location along the chromosome. So this is what you may see. This is the key part of a microarray report. And let me explain what this means. First of all, it may have these letters at the beginning. It stands for the International System for Human Cytogenomic Nomenclature. And so that just indicates to whoever's reading it that the way the data is interpreted is using standard methods for describing it. The next part, ARR, means it's an array. That tells the reader the technique that was used to create the result here. The HG19 is the Human Genome Assembly Build Number, and this may surprise you, but even though the human genome is thought was claimed to be entirely sequenced. There are places that are repetitive, that were really hard to sequence. They tend to not have genes. But what that does is, if one of those regions turns out to be, when it actually is sequenced and the data are available, it could turn out to be larger or smaller than it was guessed to be. And what that does is it changes the position and the actual length of the chromosome. And so what a microarray report is based on is the location, the number, nucleotide location from 1 to some number. And so when those numbers are adjusted a little bit, that's really important. It changes the location of a gene just slightly, and so it's really important to compare one build to another, and to know which build of the genome assembly is being described here. So that's really important. The next part is the chromosome number. Okay, that's obvious. The next part is the banding pattern. So you saw the black and white bands. They all have numbers that are assigned to them. And so this is the banding pattern affected in this microarray report, where they found something that wasn't typical. The next part are the molecular coordinates. And as I said, the molecular coordinates begin with the number 1 at the end of the top, or the end of the P-arm of the chromosome, and continue to, and you saw the chromosomes were different lengths. So this is the molecular coordinates. And so in this particular case, this deletion is from about 53.8 million to 62.6 million base pairs as the location on the chromosome of this change. And what that really is you can think about it as sort of the chromosome, the bands, and the molecular location being the country, the state, and the zip code plus 4. So it gets more and more precise as you go across reading this. And the very last thing, is the really key part, is the number of copies. And as you saw in the earlier slide, 2 is normal. So a microarray report will show you something that's not normal. So it'll be 1 copy, or 3 copies, or 4 copies, or whatever is found. So in this case, this specific region in this person, hypothetical person, was found in 1 copy. So the limitations of this information. First, again, as I've said, it doesn't tell you anything about conformation. It doesn't tell you so this particular deletion, I can tell you, hypothetical deletion is just a chunk out of the middle of the long arm of chromosome 18. So where's the end of that chromosome? Is this truly an interstitial deletion, so it's a piece out of the middle? Or is that piece that's at the end now somewhere else? So it doesn't tell you that. The other really important thing is to ask about the resolution. Many, many early microarray reports were low resolution. Still better than the chromosome study under the microscope, but lower resolution. So let me show you. So here's an example. Again, this is against the map, and let's imagine that every one of these red arrows represents a probe on the microarray. So this would be a very, very low resolution microarray. But it shows that, in fact, one of those probes is indicating there's 1 copy instead of 2. And so you would say that that deletion goes from between the red arrows on either side of the yellow to describe the potential size of that deletion. But imagine it was a lower resolution microarray. Again, only 1 probe is showing a deletion, but the red probes on either side of it are further away. So you think or believe or describe that deletion as being bigger than it really is. So the resolution is important. It's something to ask about and have explained to you, especially if you have an older report. If there's some indication clinically that it may be larger or smaller than believed, you might have justification for having a newer report done. So variations in this line across of information could be multiple chromosomes. If there are multiple deletions or duplications, this line would be repeated, describing the location and the extent of any of those chromosome copy number changes. The other thing you might see is after this line, it could say Matt or Pat. Now, if you remember, I showed you that anything you could do with scissors, glue, and a copy machine can really happen. And it does really happen. And each of us have small duplications and deletions within our genomes. So if a duplication or deletion was identified in a microarray, that's why they usually also do the parents. And then the child's report would be amended to say, if a deletion or duplication were also in a parent, it would be amended to have the mat, if it's the maternal inherited deletion or duplication, or pat for a paternally inherited deletion or duplication. In which case you would presume that it's a normal variant and not to really be concerned about it. So those are a couple of the variations that you might see in something like this. Now, what is contained in this line really dictates what's going to be found in the text. And so the things you might see in a text from a report that has this particular line would be something that says a single copy loss, or it might be described as a deletion, or it might be described as heterozygous, meaning one copy and not two has this deletion. You might also see the words interstitial or terminal. A terminal deletion means it's from the end of the chromosome that's missing. So it doesn't mean it's deadly necessarily. It's not describing the outcome. It's just describing the location of a deletion or a duplication on the chromosome, whether it's at the end of the chromosome or is something occurring, like in this example, in the middle of the arm of the chromosome. It might also say de novo. So if this is something not seen in the mother or the father, then it would be considered de novo. And if the child has medical conditions not seen in the mother or the father, and this chromosome change not seen in the mother or father, it would be thought that one is responsible for the other, or the chromosome change is responsible for the condition, and it would be called de novo because it's new in the child. The other thing it may contain is a mention of the number of base pairs. In this case, all you have to do is subtract the 62 million from the 53, and you get 8.7 million base pairs of DNA, or 8.8 megabases, the short for million base pairs of DNA missing. So it just describes the size of the change. The other thing that might be in the report are mention of the genes. Sometimes reports mention OMIM genes and sometimes total genes. OMIM genes, OMIM stands for the Online Mendelian Inheritance in Man. It started out as a book and then eventually went online. And what it is a database of what is known about all the genes for which there is something known. There are many genes that we don't really know anything about. We're not even sure if they're real genes. They might be pseudogenes. So they're hypothesized to be genes. So OMIM genes are listed because those are genes we know at least a little bit about what they do. Maybe not enough, but we know something. Total genes would be always a larger number, and those include genes about which nothing is known, or genes that may not even be real genes. So that's the difference between those. The other thing you might see in the text are words about pathogenic or benign, which now my opinion about this is it's kind of a crazy designation, and it's evolving what those terms mean. So a gene that's said to be pathogenic when you have one copy is very rarely pathogenic or causing a problem in every single person that has one copy. It may be just in most of the people who have one copy. Benign is similar in that, and so much is not known about the effects that this is why you really need to have a conversation with a genetics professional about what each of these terms mean in your particular case. The other thing that makes everyone a little crazy is when it says a variant of unknown significance. So we don't know. But the key part about this is what changes. The interpretation of this report, what it means medically, what the implications are, this is going to change over time, even if the actual data about the deletion or duplication, location, size does not change. What we know is changing all the time. This is a really vibrant area of science and medicine, and so it's important to maybe check in a few years with a genetics professional and find out more and find out if the implications of your report have changed over time, because it likely has. So how do you find a genetics professional? First of all, ask the lab that did your report. They often have genetics professionals, physicians or genetic counselors on staff who are there to help the families who received reports that identified a chromosome change. Also, you can go to the website for the National Society of Genetic Counselors and the website for the American College of Medical Genetics and find a genetic counselor or a genetics physician who can help you understand the implications of the report for you. So hopefully this information just helps you understand a little bit about what you're looking at so you feel much more prepared when you talk to a genetics professional about the implications of your specific report. So, thank you so much.